morning to everyone. I want to thank the program committee for accepting our presentation in the CIBERNED forum. As Justo said, and probably all of you know, our work in the CIBERNED has been in relation with the cannabinoid system, with the possibility that cannabinoids could be neuroprotective compounds in the context of neurodegenerative disorders. This is a consequence of the role of this system in the control of cell survival. But this has another two possibilities of therapeutic application. One of them corresponds to the possibility that cannabinoids could serve as antitumoral compounds, eliciting the apoptosis of transformed cells, and this could be important and promising in the context of cancer, particularly brain cancer, glioblastoma. The group of Manuel Guzman has contributed significantly in this area. In the other side of the coin, there will be the possibility that cannabinoids could serve as neuroprotective compounds, then increasing the survival of healthy neurons and also glial cells. And this is, of course, our interest and our promise in the field of neurodegenerative disorders. So I will present three evidence to support this potential of cannabinoids as neuroprotective agents. The first corresponds to the possibility that the endocannabinoid system could be, in fact, an endogenous protective system against different kinds of insults that may damage the brain. This has been a proposal by Rafael Mecholen in the Hebrew University based on the following observation. The first is that endocannabinoids have massively generated in response to brain damage in different experimental conditions. CB1 receptor, the typical neuronal receptor on the endocannabinoid system, is upregulated in some cases, not frequently because CB1 is present in neuron, and neuron degenerate in the case of neurodegenerative disorders, so it's expected a reduction in CB1 receptor, but in some cases there is an upregulation, and the disorder that I will present today is one of the cases in which CB1 receptor is upregulated. And if CB1 is regulated only in some cases, CB2 is specifically upregulated in most of the neurodegenerative cases. CB2 is present in microglial cells, in astrocytes, and is affected by the change that these glial elements experience in the generation of the neurodegenerative disorders. This was a first discovery by the group of Julián Romero in Fundación Hospital Alcorcón, who was the first to demonstrate that CB2 receptors are present in the microglia that surround the senile plaque in the case of Alzheimer's disease. We also demonstrated that it's also present in microglial cells in Huntington, also in astrocytes with this selective marker of this type of glial cells, and more recently we have found that it's also present in microglial cells in Parkinson's disease. This is the first group of evidence. The second evidence, this of course means that the endocannabinoid system could respond to the idea that this is a silent system that is activated in response to different challenges, and the objective of this activation is, of course, the control of brain homeostasis. The second group of evidence came from the different in vivo and in vitro model that has been used to see if cannabinoids could increase neuronal survival. Here you have a list of the different pathological conditions in which this has been investigated. And the major of these, the most important studies has been done in animal model, in cell model, but possibly the challenge for the future is to develop clinical trial as the clinical trial that Justo just presented in Huntington disease with one of the cannabinoid-based medicine that has been recently approved with the Satives. And finally, the third part of the evidence of the neuroprotective function of the cannabinoid system came from the molecular and cellular mechanisms that are activated by these compounds and that provide neuroprotection in different experimental conditions. One of the added value for the cannabinoids is that they work on different targets, and they can control different processes, and this is possibly one of the advantages compared with other kind of compounds that could cover a specific cytotoxic mechanism in the neurodegenerative disorder. Cannabinoids could be multifactorial in this context, and they can provide control and improvement in many of the different cytotoxic mechanisms that they are operating in neurodegenerative disorder. Some example of this can be found here. Cannabinoids could be anti-cytotoxic compound. They can normalize glutamate homeostasis, and it could be done by a presynaptic effect on CB1 receptor located in presynaptic glutamatergic terminals, but also in the postsynaptic neuron, in which they can control the entry of calcium and the activation of calcium-dependent destructive pathways. This would be an effect normalizing glutamate transmission, glutamate homeostasis that could be CB2 receptor are present in glial. CB2 receptor are present in glial cells. I put some example in previous slides, and in this context, CB2 receptor could control the effect of glial cells on neuronal survival through increasing anti-inflammatory mediator by reducing pro-inflammatory factors. And finally, cannabinoids could also produce effects that are mediated by cannabinoid receptor independent mechanisms. Some of these correspond to the control of oxidative stress. This is a typical effect produced by this phytocannabinoid, CBD, cannabidiol, one of the components of the sativist medicine. Cannabidiol act on the control due by its chemical structure and the control of the production of reactive oxygen species, then improving the antioxidant defense. But other possibilities has been also proposed for CBD. In this context, the effect could be mediated by a no target, at least at the moment, maybe another cannabinoid receptor type of the involvement of the nuclear receptor of the PPR family. In this context, they could add on different transcription factors, for example, the transcription factor that control the antioxidant response of the NF-kappa B that control the inflammatory scenario. So these are proposals for the mechanism of action of CBD, one of the compounds that is in our objective to be investigated in the context of neurodegenerative disorders. So our group work in phytocannabinoids. This is important. We also work in the synthesis of compounds that could be that are synthesized in the group of Pilar Goya in Instituto Química Médica in Madrid, and that could be selected of the different targets, pharmacological targets for neurodegenerative and neuroprotective therapy in these disorders. And we are trying to analyze this potential in vitro and in vivo model. Our work is basically preclinical in different disorders that are in this list. We have fluorescent more in some disorders, Huntington disease, for example. Justo just mentioned that we already arrived to the clinical trial. In other disorders, for example, the one that I will explain here, spinocerebellar ataxia, we are in this moment in the process of identifying the different pharmacological targets that could be interesting for a cannabinoid-based therapy in these disorders. As you know, spinocerebellar ataxia are part of the polyQ disorders. It's a heterogeneous group of disorders. They are all produced by an expansion of the of different genes that encode different proteins. Most of them call it ataxin. This mutated protein, they have an expansion of glutamine that is toxic for a specific group of neuronal subpopulation. In the context of spinocerebellar ataxias, the case symptom and motor incoordination and ataxia, of course, and the area most affected is the cerebellum, particularly different cerebellar neurons, and most particularly the Purkinje neurons. So our project in spinocerebellar ataxias has two main objectives. The first one is to analyze the status of the endocannabinoid signaling using postmortem cerebellar samples from scapatians. The idea of this, of the objective of this, is to identify potential targets. And the second part of our study is to work with a mutant mice for this disorder, SCA3, that is the most prevalent within the SCA disorder. In this case, we also tried to identify the change in the endocannabinoid signaling and to conduct pharmacological study to see possible beneficial effect of these compounds in these mice. The first objective, the work in the human sample, is the work that I will present here, that is completely finished, and we just started to work with the mutant mice that have been provided by a Portuguese group that work in this area also. So this is the characteristic of the different subject patterns and control that were recruited, whose samples were recruited for the study. And this is a first characterization of the change in the cerebellar cortex of the Scapatian. As you will see here, there was a dramatic loss of Purkinje neuron, typical of these disorders. Also, there is, in the same pattern, it's possible to see Purkinje cells with different level of degenerations. We also observed that the pattern has an important microglial activation and also an important astrogliosis, labeled in this case with these specific markers. Going to the cannabinoid system, the CB1 receptor was one of the cases I said before that in this case we observed a significant upregulation of CB1 receptor in the parenchyma, in the cerebellar parenchyma of the Scapatians. This happened in the Purkinje cell layers and also in the neurons in the dentate nucleus. In both cases, the levels that we quantified in the Scapatian were significantly higher than in the case of the controls. This also happened in other areas of the cerebellum with increase in the granular layer and in the white matter areas reaching the foliar surrounding the dentate nucleus. Of course, the CB1 receptor were located in the Purkinje layer in Purkinje neurons, and we did double scanning analysis to see this possibility using calvindin as a specific marker of this neuron, and we found a good co-localization between CB1 receptor and calvindin. The orthogonal reconstruction also confirmed this co-localization. CB2 receptor was, of course, located in areas that probably are very rich in glial elements. This is the case of granular layer, or also the white matter areas reaching the folia surrounding the dentate nucleus. In this case also there is an increase in patterns comparing with controls. And when we did a colocalization study to see the cellular substrate of this CB2 receptor located in these areas, we found that, of course, they correspond to astrocytes or they correspond to microglial cells, something expected with the typical profile of CB2 receptor. However, it was really surprising that we also find CB2 receptor located in Purkinje neurons. This happened also in the case of Scapatian and at a level higher than in the case of the control. This happened in the Purkinje cell layer and also in the neurons of the dentate nucleus. And when we did double labeling studies to see the neuronal substrate, we also found that this corresponds to Purkinje neuron, that they are labeled with CB2 and calvindin antibodies, and the orthogonal reconstruction confirmed this co-localization. We also worked with the two major enzymes, the two major degradative enzymes of the endocannabinoid system, the FA and the monoacylglycerol lipase, and in both cases there was also immunostaining in different areas of the cerebellum in Scapatian at levels also higher than in the case of the controls. This happened with FA and also with MAG lipase, the specific enzyme that degrades 2-AG, the second endocannabinoid. This happened in the Purkinje layer and also in the neurons located in the dentate nucleus. So in conclusion of this study, of this part of the study, we can say that both CB1 and CB2 receptor experience a significant upregulation in the cerebellar area, in different cerebellar areas in the Scapatians, and the enzymes that degrade endocannabinoids, FA and MAG, experience a similar response. Of course, this possibly is a paradoxical observation, because we found that change in receptor and we change in the enzyme that degrade the ligand of this receptor. We do not know at this moment, of course, what is the primary effect and what is the secondary. It's possible that CB1 and CB2 receptor are the primary change And as a consequence of this, enzymes respond to reduce the level of endocannabinoids, so the ligand that activates the receptor. Of course, this is impossible to be studied in the postmortem human sample, and it will be studied in the SCA mutant mice. And the third conclusion is that the presence of this endocannabinoid element, particularly the presence of CB2 receptor in neurons, open up the possibility to use this receptor and also the enzyme, of course, as a possible therapeutic target for future treatment with cannabinoids in this type of disorders. So this is the objective that was completed. The second objective has been just started. Let me only to present a few preliminary data. The objective in this case is to repeat the same in the SCA3 mutant mice to see how are the CB1, CB2, and the Fa and MAG enzymes in the cerebellum of these mice. An advantage here is that we can do a temporal analysis at different stages of the process of the pathogenesis in the case of the scapatians that was not possible in the case of the scapatians. And the second is to conduct pharmacological experiments, of course, and even the possibility to cross these mutant mice with one of the CB1 or CB2 deficient mice to see if we can obtain an aggravation or change, of course, in the pathological phenotype. This is work that is, of course, projected in the lab. We start to work with this transgenic mice for SCA3, the most prevalent SCA disorder. This has been generated in the group of Patricia Maciel in the University of Minho in Portugal. It's a progression of the past model that they generated with only, if I remember well, 95 repeats. In this case, the transgenic mice has 135 repeats and has been generated with the cytomegalovirus promoter. So this group has a good experience in this kind of experimental tools. So, and they have not published yet the observation about this, the pathological phenotype of these mice. I can advise some of the data that they have. This is the work of Patricia Maciel with a temporal analysis of the different observation at the neurological level that they found in these mice. In our hands, the mice also develop a significant change in some neurological parameters. For example, there is a loss in weight that was observed in the week 11. At week 11, there is also change in other in some motor tests, but Rotarod, that possibly is the most powerful experimental test in this case. The first age at which we observed some reduction in the Rotarod performance by this animal is at the week 11, sorry, at the week 31. We have also analyzed the brain of this animal with micro-PET analysis. We observed that we did this study at two different ages, animal of 21 week of age, so possibly a pre-symptomatic or early symptomatic time for this animal, and also at 42 weeks after birth. That is a typical age at which the animal has important neurological deterioration. And what we found that was a reduction in the metabolic use of glucose in this analysis, particularly at the age of 42 weeks after birth. And well, in this moment we have collected samples from these animals at 3 different ages, pre-symptomatic, once the pathology has initiated, and when the pathology has progressed significantly. These samples are in our freezer in the lab, and we are starting with the analysis. And we expect to have a good picture about the atrophy of the cerebellum that possibly develop in these animals, and see this in parallel with the change that we expected to find in CB1, CB2 receptor and the enzymes of the endocannabinoid system, Fa, MAG, and other enzymes. Once we have all this data, we will progress to a pharmacological study with the most appropriate cannabinoids depending on the chain of the potential targets. And I expect to be here in a future CIBERNED forum to present what this experiment has finally shown us. And let me finish with introducing people of my lab, particularly the people that is underlined in this list, that has been the people that has been particularly engaged in this study in SCA disorders, our funding agencies and collaborators around the world. And here you have a recent picture of our group. And thank you for your attention.
So thank you, Javier, for keeping the timing, and we have time for questions. Are there? Yeah, over there.
Javier, so CB2 is sort of better known as a receptor on the immune cells in periphery, and I just wondered, have you looked at all in cerebellum to see if you see inflammation, or if some of the CB2 receptor staining you see is actually on microglia?
In the patients, we did analysis with two markers for glial elements, IBA1 and GFAP, and we found important level of inflammation. But all the work was done in the cerebellum, not in the periphery. I imagine that it's possible that CB2 receptor, for example, is located in peripheral immune cells, and it's a good marker of this, and it's possible in some cases that there is activated immune cells that can cross the blood-brain barrier and infect the brain and to produce some presence of CB2 receptor in the brain. This has been done in other disorders. I don't know in this case.